So this is one of the things that people have been asking me about.
You
So
rotate that a tiny bit. No, like this.
this way towards you guys.
Yeah.
I know.
That's better. It was picking up the glare.
The thing's hard to see either way. I should have done a different color.
It's better.
I just.
Oh, you're recording. I'm sorry.
No, you're fine. That's why I'm doing like this. Is to try to record it so people can like do this without having to ask me a million things. But one of the questions I got from people was like, how much are we like going into the weeds with like macromolecules versus like not? I just did the nucleic acids and the lipids on this because he kind of spent more time separately on sugars and amino acids. So from what I can tell in terms of like what people really wanting for like macromolecules for like nucleic acids, you're just wanting us to know like basic structure of your nucleic acid, which is your phosphate group plus some ribose sugar, which is just a 5-member ring, so it's oxygen and 4 carbons, and then some sort of heteroatomic base. So like I drew like a couple of different variations. So this one's like cysteine and adenosine, and then this one is the difference between higher. I don't know the nucleic acids. It's on our thingy. I don't think we actually have to know these, but the idea is that this is your base right here, which goes right here on that ribose sugar. So it's going to take the place of that OH group that's on this sugar. So like off of this aromatic carbon, that's where that base is going to go on this ribose for your DNA or RNA. And then you have a phosphate group, which I just wrote PO because I'm lazy, but that's the idea that it's like that PO3, and then you have like bond into the oxygen or whatever. So that's a phosphate group. And so this is the entirety of structure for a nucleic acid. So that's the extent of what he wants us to know. It's phosphate, ribose sugar, which is a 5-member sugar with 1 oxygen and 4 carbon. And then the fact that there is a heteroatomic base. I don't think he wants us to memorize the bases, but I'm not the teacher, so I don't know what the expectation is for Dr. Selfrey. And then he wants us to know like functions of macromolecules. So basic functions of nucleic acids is very simple: transcription, translation. Probably wants us to know the difference. Maybe did he mention anything about that? I don't think he did. I mean, it still could be a question like that's very, very basic. Like we learned that whenever we do like bio or biochem and whatever, like what the difference between like transcription and translation is. So it could still be on the test, but I don't think it will be. Okay, hold on. Okay, so next macromolecule that we kind of talked about was lipids. We actually had the lipids before we talked about nucleic acids, but I just did them in this order because I knew I was going to need more room for lipids. Same basic thing is I don't think he wants us to know like the eicosanoids. He doesn't want us to know like specifics about these. He just wants you to know the general structure of what these things look like. So your fatty acids are these really long carbon chain tails with some sort of carboxylic acid. You can have other things off of this, you know, in terms of like whenever he showed us like arachidonic acid and then showing us making those eicosanoids off of it. The general idea is that, you know, like they start with something that looks like this and they change it to make it some sort of signaling molecule, structural molecule, or storage molecule. So arachidonic acid is storage and membrane. So it's stored in the membrane, but it also works as like a storage mechanism for all the hormones that we make. And then once it's converted into those other structures, it's used for signaling. So fatty acids kind of serve all 3 purposes in terms of like structure, storage, and signaling.
Can you go into saturated versus unsaturated with the bonds?
Yeah. So the idea of saturated versus unsaturated. So when you say something is saturated, you're specifically talking about hydrogens. So if you have something that has a bunch of hydrogens, it is saturated with hydrogen. Versus if you have double bonds thrown in, like you have double bonds all along this structure here, then you're basically removing hydrogens in order to make carbon-carbon double bonds. So then you're unsaturating that molecule. So that's all that saturation has to do with, is how many hydrogens are on that chain. And you only really use that when you're talking about like fatty acids. So yeah. I don't know how much he wants us to know in terms of like the number. They did mention 10 to 22 carbons for that tail. That could just be a very easy like point question, where you'd be like, you know, this has like a fatty acid has how many carbons in it. I feel like that could be a very easy question to ask for points, and it's like, I don't know, then, you know, that's points you could lose. But yeah. Next lipid that he went into was glycerides. He didn't really talk about them too much, from what I remember in that lecture. But just knowing that it's a glycerol, which is a 3-carbon ring that has these OHs on it. So it's an all, so it has OH groups. So this is basically an alcohol that is a good structural alcohol, because then it can build these fatty acids off of this glycerol molecule. So that's what this is you can have any, I put different R's, basically to signify you can have any different fatty acids off of this glycerol molecule, and this is a glyceride. You can also phosphate your glycerides, so you have a phosphoglyceride, which is kind of the general structure of how we have like our phospholipid bilayers, things like that. So that's where the whole like, you know, you have your phospholipid head and you have your fatty acid chain tail. That's where that kind of comes from. These are pretty much just storage of energy. That's our like secondary like energy mechanism. But they do sort of serve like membrane structure as well. But it's more of just an idea of storage. Like typically using alcohol for storage. Number 3, any questions on the glyceride stuff before I go on? No?
No.
Okay. Number 3 was non-glycerides. He also generally spent a whole lot of time on this. I don't know how in depth he really wants us to know it. I think more just knowing that those non-glycerides are for signaling and structure. I don't know if we're expected to know the structures of these. I don't think it's terrible to expect it, but I think it's more just knowing that he's going to say, here's a sphingolipid. What is that going to be used for? And that's going to be used for signaling. Same thing for steroids. What are they used for? Signaling. Waxes are more structure. So that was where he talked about the bugs and their like exoskeletons and stuff like that. So the idea is that you have this ester group. So like it's one of the functional groups we learned where it's like carbon and then you have your carbonyl group and then oxygen and another carbon or whatever. So your idea is, you know, you can have any chain of carbons on the other side of this ester group, and that creates that that shell, that exoskeleton for insects. I don't remember if he said if we have any like waxes that we make in our body, but I can't imagine it's impossible. I'm sure there's some structural component of like our membranes or something. But yeah, that's pretty much it. I would say if you wanted to like memorize this sphingolipid thing, just know that it's got an amide group on one end, and then it's got basically like a double bond with an OH on the other, and then just an ether group on the top. I don't think you necessarily have to memorize like anything specific. I think it's just the idea that like it just looks like that, but I'm pretty sure it's just going to be sphingolipid signaling. And then the last one is your complex protein, complex lipids, and that was your like glycolipids and your lipoproteins, which is just literally like lipid plus protein and lipid plus sugar. And we've kind of already talked about those in like AMP and farm and all of that, with the idea that your lipoproteins are going to be storage and signaling, kind of a little bit of both, but more storage than anything else. And then you have glycolipids, which are going to be signaling and structure. So think about like a glycolipid, you know, layer on your membrane and how that's used to like identify your white blood cells, whether or not the cell is healthy. Whenever you talk about like lipase in whichever class it was, we're talking about lipase. Is that AMP? It all blurs together at this point for me. So, yeah. What questions you got? I feel like this is pretty straightforward. I think a lot of people are getting into the weeds of this in terms of like really going into it. I don't think we need that. I mean, I could end up getting a terrible grade because of that thought, but the fact that he didn't really go into it, and from the standpoint of him just wanting, like saying in his like thing that he really wanted basic things, I think this is the extent of what he wants in terms of macromolecule stuff and function. So nucleic acids, transcription, translation, lipids, structure, storage, and signaling.
Did you talk about fatty acids being sister trans at all?
I did not. I don't.
Did he?
Did he?
He mentioned it, but I'm like
So.
saying that the trans is more stable compared to the sisters.
So all that has to do is it's only dealing with your unsaturated fats, and it's whether or not it does this or this after the double bond. So we'll love
He
this.
didn't even put that out. He just found out sister trans.
Yeah.
So I'm
So
like, I don't know.
that is more related to the idea of like E or Z, but like in terms of like when you're doing it with like fatty acids, you call it cis or trans. So the idea is they're on the same side, it's cis, so it's less stable, but it's easier to break down, which means our bodies can actually use it, versus trans are more stable because they're, they don't have that tension, basically. And the idea that these are less healthy for us because of the fact that it's harder for our body to break down something that's stable. So that's all that was about. There's not really a whole lot to read into it because he didn't really talk about like identifying cis or trans fatty acids or anything as far as I can remember. Once again, I make mistakes, so I could be totally wrong, but
He just brought up
yeah.
about the being stable.
Yeah. Cool. I'm gonna hit stop on my recording.